Embryo and gamete. Cryopreservation. Techniques for freezing and thawing eggs, sperm, and embryos. Cryopreservation is the process of freezing and storing biological materials like eggs, sperm, and embryos for later use. This technology has revolutionized fertility preservation, allowing individuals to delay reproduction or save reproductive material for future IVF cycles, medical treatments like chemotherapy, or genetic testing. Embryo and gametic cryopreservation have become essential tools in assisted reproductive technology, ART. Proper freezing and thawing techniques are critical to ensure the viability and functionality of the gametes, eggs, and sperm, or embryos after thawing. Below is an in-depth look at the techniques for freezing and thawing eggs, sperm, and embryos. 1. Techniques for cryopreserving sperm. A. Sperm. Cryopreservation, freezing sperm. Sperm cryopreservation involves freezing sperm for future use. This can be done for fertility preservation prior to chemotherapy or to store sperm for ART procedures like IVF or ICSI. 8.1 Sperm collection and preparation. Collection: sperm is collected through masturbation or testicular aspiration if there are issues with ejaculation. Processing: the collected sperm is mixed with a cryoprotectant solution to protect it from ice crystal formation during freezing. The cryoprotectant, for example glycerol, prevents cell damage by reducing ice formation inside the sperm cells. Separation: sperm is often processed to isolate the best quality sperm through density gradient centrifugation or swim-up techniques, improving the chances of successful fertilization later. 8.2 Freezing process. The sperm sample is placed in small vials or straws, and the vials are slowly cooled to temperatures of minus 196 degrees Celsius, liquid nitrogen, for long-term storage. Controlled rate freezing is typically used to prevent the formation of large ice crystals, which can damage sperm cells. B. Sperm thawing. Thawing sperm is a delicate process that must be done carefully to maintain sperm viability. The sample is gradually warmed to body temperature, and the cryoprotectant solution is carefully removed to avoid osmotic shock to the sperm cells. The quality of sperm after thawing can vary, and motility and morphology are often assessed before use in ART procedures like IVF or ICSI. 2. Techniques for cryopreserving eggs. A. Egg cryopreservation, freezing eggs. Egg cryopreservation is used to preserve eggs for women who want to delay childbirth or those facing medical treatments like chemotherapy that can affect fertility. A1. Ovarian stimulation and egg retrieval. Women undergoing egg freezing usually undergo ovarian stimulation to produce multiple eggs. This is followed by egg retrieval under sedation, where mature eggs are collected from the ovaries. 8.2 Freezing process. After retrieval, eggs are placed in a cryoprotectant solution to protect them during freezing. The cryoprotectant helps prevent ice formation inside the eggs, which can lead to cell damage. The freezing of eggs typically uses the vitrification method, a fast freezing process, which avoids ice crystal formation by rapidly cooling the eggs and solidifying them into a glassy state. Vitrification is more successful than traditional slow freezing methods in preserving egg viability. B. Egg thawing. The thawing process is critical and must be done carefully to avoid damaging the eggs. After thawing, the eggs are checked for survival, and those that survive the thawing process can be used for fertilization via IVF or ICCSI. The thawed eggs are often assessed for morphology and maturation, and only mature eggs are used for fertilization. 3. Techniques for cryopreserving embryos. A. Embryo cryopreservation freezing embryos. Embryo freezing is a commonly used procedure in ART, where embryos that are not used in the current IVF cycle are frozen for future use. A1. Embryo culture. After fertilization, embryos are cultured for 3 to 5 days to the blastocyst stage. Embryos that are not selected for transfer are frozen, especially those that are high quality and have a high potential for implantation. A.2. Freezing process. Embryos are frozen using vitrification, which involves rapidly cooling the embryos to prevent ice crystals from forming inside the cells. This method has a higher success rate compared to traditional slow freezing. Cryoprotectants are added to the embryos before freezing to protect them from ice crystal formation, and the embryos are stored in liquid nitrogen at negative 196 degrees Celsius. B. Embryo thawing. The thawing process of embryos is also done with care to ensure high survival rates. Vitrified embryos are thawed gradually and transferred to a warm culture medium. After thawing, embryos are checked for viability and quality before transfer into the uterus. Frozen embryo transfer FET cycles have similar or sometimes higher success rates compared to fresh cycles, particularly when embryos are frozen at the blastocyst stage. 4. Challenges and success rates of cryopreservation. A. Success rates. The success rate of thawed embryos is generally high, especially when embryos are frozen at the blastocyst stage. The success rate for thawed sperm and eggs is lower compared to fresh samples, with sperm and embryos typically having a better thaw success rate than eggs. The success rate of egg freezing has improved with vitrification, and women who freeze their eggs at a younger age have a higher chance of success when using thawed eggs later. B. Challenges in cryopreservation. Sperm cryopreservation. The main challenge is maintaining sperm motility after thawing, especially if the sperm quality was compromised before freezing. Egg cryopreservation. Eggs are more sensitive to freezing and thawing than sperm or embryos, and their survival rate after thawing may vary. Eggs that have not matured properly prior to freezing have a lower chance of fertilization after thawing. Embryo cryopreservation. While embryos typically have better survival rates post-thaw than eggs, freezing and thawing may still cause cellular damage, particularly if not properly handled. 5. Conclusion. Cryopreservation of eggs, sperm, and embryos is a vital component of modern ART. Vitrification techniques have improved the success rates of cryopreservation, particularly for eggs and embryos. However, the process still requires careful attention to detail and expertise in freezing and thawing techniques to ensure the highest chance of success when using thawed material in future ART cycles.